Cell walls. Because of lipid protein plasma membrane of less than 10 nm thickness can be expected to offer only minimal protection for a cell's contents, it is not surprising that naked cells are extremely fragile structures. The cells of nearly all organisms other than animals are enclosed in a protective outer envelope. Protozoa have a thickened outer coat, whereas bacteria, fungi, and plants have distinct cell walls. We will restrict our discussion to the cell walls of plants, which were the first cellular structures to be observed with the light microscope. Plant cell walls perform numerous vital functions. As discussed earlier, plant cells develop a turgor pressure that pushes against their surrounding wall. As a result, the wall gives the enclosed cell its characteristic polyhedral shape. In addition to providing support for individual cells, cell walls serve collectively as a type of skeleton for the entire plant. In fact, a tree without cell wall might resemble in many respects a human without bones. Cell walls also protect the cell against damage from mechanical abrasion and pathogens, and they mediate cell-cell interactions. Like the ECMs at the surface of an animal cell, a plant cell wall is a source of signal that alter the activities of the cell that it contacts. Plant cell walls perform numerous vital functions. As discussed, plant cells develop turgor pressure that pushes against their surrounding wall. As a result, the wall gives the enclosed cell its characteristic polyhedral shape, in addition to providing a support for individual cells. Cell walls serve collectively as a type of skeleton for the entire plant. In fact, a tree without cell wall might resemble, in many respects, a human without bones. Cell walls also protect the cell against damage from mechanical abrasion and pathogens, and they mediate cell-cell interactions. Like the ECM at the surface of an animal cell, a plant cell wall is a source of signals that alter the activities of the cells that it contacts. Plant cell walls are often likened to fabricated materials such as reinforced concrete or fiberglass because they contain a fibrous element embedded in a non-fibrous gel-like matrix. Cellulose, whose structure was described on page 47, provides the fibrous component of the cell wall, and proteins and pectin, described below, and provide the matrix. Cellulose molecules are organized into rod-like microfibrils that confer rigidity on the cell wall and provide resistance to tensile forces. Each microfibril is about 5 nm in diameter and is typically composed of bundles of 36 cellulose molecules oriented parallel to one another and held together by hydrogen bonds. The plant cell wall, electromicrograph of a plant cell surrounded by its cell wall. The middle lamella is the pectin containing layer situated between adjacent cell walls. Electromicrograph showing the cellulose microfibrils and hemicellulose cross-links of an onion cell wall after extraction of the non-fibrous protein pectin polymers. Schematic diagram of a model of a generalized plant cell wall. Hemicellulose is there, cellulose microfibrils is there, glycoprotein is there, pectin will be there. The walls of many plant cells are composed of layers in which the microfibrils of one layer are oriented at approximately 90 degrees to those of adjacent layers. Cellulose molecules are polymerized at the cell surface. Glucose subunits are added to the end of a growing cellulose molecule by a multi-subunit enzyme called cellulose synthase. The subunits of the enzyme are organized into a 6-membered ring or rosette which is embedded within the plasma membrane. In contrast, materials of the matrix are synthesized within the cytoplasm and carried to the cell surface in secretory vesicles. The matrix is highly complex. Hemicellulose are branched polysaccharides whose backbone consists of one sugar such as glucose and side chains of other sugars such as xylose. The matrix is highly complex, requiring hundreds of enzymes for its synthesis and degradation. The matrix of the cell wall is composed of 3 types of macromolecules, which are hemicellulose, pectin, and proteins. Hemicellulose. Hemicellulose are branched polysaccharide whose backbone consists of one sugar such as glucose and side chains of other sugars such as xylose. Hemicellulose molecule bind to the surface of the cellulose microfibrils, cross-linking them into a resilient structure network. Pectins are a heterogeneous class of negatively charged polysaccharides containing galacturonic acid. Pectins are heterogeneous class of negatively charged polysaccharides containing galacturonic acid. Like the glucosaminoglycans of animal cell matrices, pectins hold water and thus form an extensive hydrated gel that fills in the spaces between the fibrous elements. When a plant is attacked by pathogens, fragments of pectin released from the wall trigger a defensive response by the plant cell. Purified pectin is used commercially to provide the gel like consistency of jams and jellies. Proteins whose functions are not well understood mediate dynamic activities. One class, the expansins, facilitate cell growth. These proteins cause localized relaxation of the cell wall, which allows the cell to elongate at that site in response to the turgor pressure generated within the cell. Cell wall associated protein kinase span the membrane, plasma membrane, and are thought to transmit signals from the cell wall to the cytoplasm. The percentages of these various materials in cell walls are generally variable, depending on the type of plant, the type of cell, and the stage of the wall. Like the extracellular matrices of animals, tissues, plant cell walls, and dynamic structures that can be modified in response to changing environmental conditions. Cell walls arise as a thin cell plate that forms between the plasma membranes of newly formed daughter cells following cell division. The cell wall matures by the incorporation of additional materials that are assembled inside the cell and secreted into the extracellular space. In addition to providing mechanical support and protection from foreign agents, the cell wall of young undifferentiated plant cell must be able to grow in conjunction with the enormous growth of the cell it surrounds. The walls of growing cells are called primary walls, and they possess an extensibility that is lacking in the thicker secondary walls present around many mature plant cells. The transformation from primary to secondary cell wall occurs as the wall increases in cellulose content and in most cases incorporates a phenol containing polymer called lignin. Lignin provides structural support and is the major component of wood. The lignin in the walls of water conducting cells of the xylem provides the support required to move water through the plant.